Okay. Let's.
Hey, I know.
Oh, I actually have a picture of one of those for you.
You'll talk about more of it.
Okay, let's do virology. We do have a virologist in the class, so if I make any mistakes,
Cool.
say no, you're wrong.
Yes, sir.
I do my best with virology. I am super interested in viruses. I find them fascinating. I think they're incredibly interesting. You'll have to knock. Close the door. I find them incredibly interesting, and yet I don't pretend to be a virologist expert. So let me know if I'm wrong. Okay. So viruses are probably the most simple, arguably living thing. Whether they're living or not, that is debatable. We'll go through some terminology. The virion, okay, so if you see the word virion, that's the infectious particle. So what would a virion be made of? Like, let's think on a molecular level what these things are made of. What are in my classes, I often teach and focus on, there's sort of 4 types of molecules that make up living things. What are they? What are like the 4 main types of molecules that make up living things? Don't answer yet, people who know for sure. Well,
Envelope.
no, like types of molecules. Think biochemistry.
DNA. Protein.
Okay, so proteins, yes, proteins. You said DNA, but there's a broader term.
Nucleic acid.
Nucleic acids. What are the other ones?
Lipids.
Lipids, and then what's the last one?
Some of them.
Sugars?
Yeah, sugars, carbs. These are kind of like the broad 4 chemicals, biochemicals that make up living things. So when you describe like a virion, which of these things can you find in a virion?
Nucleic acid.
Where would the nucleic acid be?
Inside the...
So nucleic acids will get packaged inside. Yep, so they definitely have nucleic acids. What else?
Proteins.
Is it called a protein envelope?
Capsid.
Yeah, it's more like a capsid.
Nucleocapsid.
So the proteins form like the structure or the capsid of the virus or the virion. So there's proteins, nucleic acids. Are there these other things?
So there's the envelope that has the lipids and the glycolipids
Yes.
that are surrounded, and usually they take those from the cell membrane of the host.
Very good.
Not all the viruses have that.
Very good. So some viruses have a lipid outer layer and outer membrane. They're enveloped, quote unquote. Other viruses are not enveloped. So some have lipids, some do not. Do you find carbs in viruses?
Some glycoproteins.
Yes, glycoproteins. So there's glycoproteins, which are proteins that are glycosylated. So it means you take a protein and they take a sugar and they stick it together. And oftentimes you find glycoproteins like sticking out of like the surface of the capsid. Why would you find that? What are the glycoproteins often important for?
Finding the host cells.
Yeah, so like finding the host cells. So cells can't, viruses and cells can't see, and they can't, I guess, like perceive in that sense of sight, but they can kind of like feel. They kind of have this feeling ability, and that's communicated through the glycoproteins, where the amino acid structures and the carbohydrate structures can have chemical interactions that preferentially bind with certain cell types. And that's how viruses evolve and learn to infect certain tissues versus others, is by developing or evolving this sort of like sensory mechanism on the outside of their virion. So this is a virion, and this thing is completely capable of invading a cell and replicating. Okay, any questions about that? So I kind of call viruses, they're like these packages with the cream filling, and the cream filling is like the nucleic acids, and that's an important part for understanding viruses. Is there's lots of diversity in how viruses organize their nucleic acid genome, and that's often a descriptive characteristic of the particular virus. Okay, this word, phage. What's a phage?
A virus that infects bacteria.
it's a virus so it comes from bacteriophage. So it's a virus that only infects bacteria. Okay, so what we're going to be discussing is not going to be quote-unquote phages. We're going to be discussing other viruses like flaviviruses and arboviruses and alphaviruses and Bunya viruses, but they're not phages. So there's a distinct difference. And viruses can be very specific with what they invade, what types of cells they invade. So are you going to get sick from a bacteriophage? No, because that bacteriophage has specifically evolved to infect bacterial prokaryotic cells and replicating prokaryotes. And so it's actually more likely that a bacteriophage would be your friend if it's killing like bad bacteria. So just know that viruses evolve and become very specialized to infect the cells that they infect. Let's see. Everything has viruses, eukaryotes, prokaryotes. Let's see. Okay, so let's talk more about this structure. These, the proteins that form this, they are definitely called capsid proteins. We went through some viruses have a lipid membrane. Daniel said, which is true, that the source of this membrane can come from different sources. So somebody other than Daniel, where do you think that this lipid bilayer in an enveloped virus comes from? Can a virus make its own bilayer?
Does it come from the?
No. So first of all, viruses, like, they can't make their own stuff. So where does it come from?
Is it from like a cell that it hijacked?
Yeah, but where?
Host.
But where in a cell? Cells are like big things that have lots of different rooms. Okay, so let's backtrack. If we have a eukaryotic cell, where are all the places you find membranes?
The Golgi?
You can find membranes in the Golgi. Where else?
Is it the ER?
You can find membranes in the ER. Where else?
The rough ER.
Okay, ER.
It should be on all the organelles.
There's a membrane separating the nucleus. There's a membrane in the mitochondria. There's a membrane on the outside. Like, there's membranes everywhere. My point is, viruses are different, and depending on how they've evolved, they can steal membranes from different sources. So maybe a virus might get membrane from the Golgi because maybe it replicates and forms virions in the ER, but then maybe it gets sort of moved to the Golgi and then trafficked out of the cell, and in that process maybe it acquires some Golgi membrane. Or maybe you can do what's called budding, where the virus, as it buds out of the cell, steals a little bit of the lipid bilayer. That would be an envelope that comes from the outer membrane. Okay, so just know that like viruses can take membranes from different spots, and these membranes are different, so they're going to look differently as they come out based on like what things they stole, if that makes sense. Yes? Okay. Let's see. Okay, so let's go through. Again, so I have to sort of do, some people may find this more rudimentary, but there are other people in this class who have never had any lectures on viruses before. So I need to kind of be very basic at first steps. So viruses, it's always kind of like questionable whether they are alive. So what are some characteristics of living things? And let's go through, like, let's think about viruses. So what are... Respiration, or maybe you'd say like metabolism.
Sure.
So like metabolism. What do you mean by metabolism?
Well, when I think of something's alive, I think of like a living, breathing being with blood going through it.
So like acquiring nutrients, and a lot of what goes with metabolism is growing,
Yes.
right? So like growth, even cells grow, growth and like acquiring energy and using energy. Do viruses do this?
Not exactly. Not really.
I'd say no. Like, they're definitely like, they don't grow. The virion is a distinct, very set size. That doesn't change except through evolution. So they can potentially change size by mutating their genome and changing their protein structure, but they won't grow and change their size in the life cycle of a single virus. They're not going to acquire energy in a sense of like a bacteria will actually like actively transport in nutrients and break them down and use them to build things. A virus is not going to do that. Using energy, that would be maybe where you'd be like debatable. Like they have proteins that will steal energy, but they're not going to be sort of acquiring nutrients, breaking them down, and then using them. Okay, so I'd say no. They don't do metabolism. What else are essential things that make you alive? Replication. That is like the selfish gene theory, like everything that is alive, it is its entire purpose. The only reason you are alive is to replicate your DNA. That's kind of maybe that's not the best philosophical view, but that's essentially like everything in biology, that's all they care about, is replicating themselves. So do viruses replicate?
Yes.
Yes, they definitely replicate, but what's the catch?
They hold the host.
What?
Host.
Yeah, they can only replicate inside of a host using host machinery. So they don't contain a self ability to replicate. They're sort of like parasitic replicators. But in that sense, they're very interesting because some of the earliest theories of how living things came about is from an evolutionary process starting on a molecule that can replicate. And so viruses are very interesting in that they are kind of like these little tiny machines that don't do anything really except figure out how to like replicate, although they can't replicate themselves. What else? What else do living things do?
Excrete waste, or would you say that's metabolism?
That's kind of like associated with metabolism, yeah.
I don't know if it has to be something with genetic material evolution or genetic changes or.
Yeah, I would say like adaptation, or evolution. So adaptation, you can think about this in two different ways. You could think about like an organism like us, or a monkey, or an upper eukaryote, or a multicellular organism. They can sense things in their environment, and then they can change their morphology or their behavior to adapt to the environment. Viruses cannot do that. Like a virus is a little machine, and once it's built, that's what it does. But they can adapt and evolve because they are encoded by a nucleic acid sequence, and that nucleic acid sequence can change, and therefore they can adapt. So they can adapt and evolve, but only intergenerationally, if that makes sense. They are not gonna, or not intra-generationally. So I would say yes, they do this. Let's see, what else?
They're like composed of biomolecules, like if being composed by biomolecules is an axiom for every living organism.
Yeah, you could say that. That's decent.
Nucleic acids and
So
lipids, carbohydrates.
They're composed of the four sort of like types of biomolecules. So you could say yes. They're very very close to things that are alive. So like this is why I kind of think they are kind of like alive, because if you actually really think about it, like they do a lot of stuff. Okay, so that gives you sort of like a sense of what they're doing. Okay, so let's talk about their life cycles. Okay, so the life cycle of a virus. What's the first thing that needs to happen? Invasion. Okay, so invasion is going to rely on protein-protein interactions at the membrane or the envelope or the capsid interacting with. So if you have some virus, I'm just drawing like geometrical structures because that's what they look like. And if you have some cell, whatever it is, there's going to be receptors on that cell, and they're probably going to be glycoproteins or some kind of like receptors on the edge of the virus. And there's going to be some kind of interaction. And oftentimes this interaction then induces some kind of like, I don't know if the best term would be phagocytosis, but endocytosis.
Receptor-mediated endocytosis.
Receptor-mediated endocytosis. So endocytosis, which is good, endocytosis, which is going to be an invasion or invagination of the membrane. So the membrane's going to do something like this, okay, based off that signal, and then it's essentially going to like absorb the virus. Okay, so that's typically how viruses are like invading. In a bacteriophage, they got like this weird apparatus where like everybody's seen the pictures, it's like a spaceship, they will like land on the bacteria and then they will like inject their DNA in. So there's different ways to invade, but they all have to invade. That's always step one. Okay, so what's step two?
Uncoding.
Coding?
Uncoding.
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 Oh, I see what you're saying. I would guess I would consider that
Or
kind of like.
Or it could be invasion, internalization, and then uncoding. Like it does not apply. This is only for the classic virus.
Yeah.
But like when they invade, they become internalized and they uncode.
Yes. And then they somehow get their genomic sequence out into either the cytoplasm of whatever the cell. I would kind of consider that as like a subpoint of invasion. Then what happens once they've sort of uncoded and released their information?
It depends when they're going to replicate. If they're RNA viruses in the cytoplasm, if they're
Yes.
DNA, they're going to be in the nuclei.
So in a broad sense, so you're absolutely right. In a broad sense, the next thing that needs to happen is replication. And how they do that is different for each virus, like how they get to that point. And that depends on the type of the virus and what the, I guess, genetic structure of that virus is. But the second thing that needs to happen on a broad sense is replication. And to think about how this needs to happen, when cells replicate, what's the thing that they actually need to replicate? What's the biomolecule well, they need to replicate all their biomolecules, but the blueprint needs to get replicated, right? How does DNA get replicated?
Protein?
What protein?
Ribosomes.
That's translation. That makes proteins. How do you replicate DNA? What protein? What?
Polymerase?
Yeah, it's a polymerase. And it's going to be a specific type of polymerase. So there's different types. There's DNA polymerases and there's RNA polymerases, right? So replication of viruses is going to involve some type of like polymerase. So they're going to have some type of enzyme that's going to be able to replicate their genome. And typically what happens is they essentially replicate their genome. Another question is, what enzyme causes transcription in cells?
What was the question?
What enzyme in cells causes transcription?
Isn't it also polymerase?
Yes, it's also a polymerase. It's not a trick question. Essentially, like polymerases replicate nucleic acids, okay? Except one that does transcription is an RNA polymerase, and if you're replicating DNA, it's a DNA polymerase, or if you're replicating RNA, it can be a DNA-dependent RNA polymerase, stuff like that, etc. Essentially, the point is, they need to get their genome replicated, and they also need to make proteins to form the virion. And the way that you make proteins and the way that viruses make proteins in the cells is the same way that you make proteins in your cells, because they steal your machinery. So they need to make RNA, which then can get read by the ribosome and translated into proteins. So there's always like a process of taking the genome, turning it into some form of readable RNA. What is the readable form of RNA?
RNA messenger.
Yes, messenger RNA. How do you describe the structure of messenger RNA?
Capped and polyadenylated.
Yes, for like eukaryotes, but that's more for like stability. I'm talking in terms of like readability.
They have a stop and an initial codon.
So definitely, there's going to be, if the sequence, there's definitely going to be like a start codon, there's going to be a stop codon, and what is this direction?
3 to 5.
No.
Oh, by that 5 to 3.
5 to 3.
Yes, 5 to 3.
Don't roast me.
I will definitely roast you. No, this is all for review. This is actually important, 5 to 3, and this becomes very important for like virology, because if you don't recall from your biochemistry classes, nucleic acids have direction. They're directional molecules. So when you think of 5 to 3, that's called sense. That's going in like a distinct direction, and it means that it makes sense to the ribosome. The ribosome can just take this and make a protein. If you have RNA that's antisense, let's say it's 3 to 5, can the ribosome read that and make protein? No. So if you're a virus and your genome is encoded in 3 to 5 antisense RNA, you have a conundrum, a paradox to fix. which is that you might inject your molecule, your nucleic acid, into the cell, but the cell can't do anything with it, it can't read it. So what do you think needs to happen if the virus has a weird antisense RNA genome?
A reverse transcriptase.
It needs to get translated. Yeah, essentially it needs to get, like, you need to make a different copy of it that can be read. And so there is a special enzyme called reverse transcriptase, which is also very important for virus biology. And reverse transcriptase is just another, it's a special polymerase. Okay, so reverse transcriptase, there's different types of reverse transcriptase. If it's a, let's see, if it's an RNA dependent DNA polymerase, that's one type of reverse transcriptase. So it takes RNA and it can make DNA. There's also an RNA dependent RNA polymerase, which can be also called a reverse transcriptase. And so if you were a virus and you injected this, typically they also have a protein, which is a reverse transcriptase, which is encoded inside the virion. It's built into the virion. And as soon as they inject this, their reverse transcriptase will read it and it will make a 5-to-3 complement. And then this thing can immediately get made into proteins. And the proteins cause a feedback loop where they take these molecules and make more and more and more and more and more. And then eventually you get like virus virions. So replication is the process of sort of like replicating the genetic contents of the virus and making and assembling virions. Okay? Okay, what's the third thing that needs to happen then?
Assembling.
After assembly, assume assembly is sort of like a subcomponent. We will talk a little bit about assembly, but assume assembly is sort of like a subcomponent of replication. They escape. They need to like get out. They need to get out of the cell because at some point the virus sort of uses up all the nutrients of the cell, or maybe it's attenuated and it just needs to get out and go on to a different place. So they escape. And there's different ways to escape, okay? So one strategy, I'll leave this. One strategy to escape is budding. What happens in budding?
Take pieces of like the cell membrane.
Yeah, budding is kind of like a peaceful way to leave. So if you have a eukaryotic cell and you have a virion, budding means like the virion will just go to the edge and it'll steal a little bit of the membrane and it'll just kind of like bud off. And it won't kill the cell's still alive, but the virus escapes. And then this virus goes to other cells, invades other cells, replication happens, and budding continues. That's like the peaceful way to leave. Okay, what's the other way to leave? Lysis. Okay. So lysis is where the virus perhaps is so pathogenic to the host that it doesn't know how to control itself, and it replicates so efficiently and so profusely that the cell literally becomes a factory of virions, and at some point the virions cannot fit in the cell, and they can either induce lysis or they will physically break the cell open, and then at that point the cell bursts and releases all the virions, and then they go do it again and again and again. So this is going to be quite a pathogenic virus, and so something that's more attenuated or that's evolved to become a little bit of a lesser pathogen is perhaps going to do more budding. Something that is very pathogenic is perhaps going to do more lysis, where it's literally like killing your cells. That's a bad deal. Okay. And then there's sort of like a third. It's not an escape, but it's sort of like a terminal life strategy. What's the third thing that contrasts with lysis?
Cytomegalomy.
No.
Induce,
Is that like fusion of cells?
yes, like induce like the cell increase size.
They can do that, where like, yeah, it causes a cell to fuse another. That's interesting. But that's not the one I'm thinking of. Lysis is often a counterweight to another life strategy.
It'll go dormant, won't it?
Yeah, what's that called?
Senescence.
Well, that's like cellular dormance.
Oh.
What is it called when the virus?
Latency.
If this is the lytic phase, lytic versus lysogenic. I always get these confused.
Because they're flipped, aren't
Yeah, because I always think lyse, I always think, like, so just so you know this is wrong, I always think that lyse means to lyse the cells. It's opposite of that. Lytic means to lyse the cells. Lysogenic is sort of like a dormancy. Lysogenic. So what happens in the lysogenic phase of a virus?
It implements itself into the genome.
How does it, so, okay, so that's exactly what happens. These are actual words you need to know, distinct semantic words. It's called integration. Integration means it essentially like recombines into the genome. So if you've ever heard like in your body, you have cells that have DNA, and in those DNA, you have viruses that have integrated into your genomes. You are the carrier of like all kinds of viruses that have integrated into your genomes. It literally means like if you have a chromosome, a virus genetic sequence has literally inserted into your genome. And now every time you replicate, guess what else you're also replicating? That foreign body that is now in your DNA sequence. So this is like, lysogenic phase is like a clever way that the virus can get replicated without doing any work. It's just sort of like a parasitic DNA sequence. It inserts itself, and then every time you replicate, you just replicate that thing also. So viruses can switch back and forth between lytic and lysogenic. So they have sensing mechanisms where there's some bacteriophages where when it senses the cell becomes inhospitable, it will then induce and switch back to the lytic phase, and it will kill the cell and pop out and try to get somewhere else. And then if it's a good place to be, it'll just sit in lysogenic phase. So viruses can switch back and forth. It can get complicated. You had never heard of lysogenic phase before?
Me?
Yeah.
No, I've heard it.
Oh, you have heard it. Okay. I thought you said you had not. Okay.
No, where'd you get that idea?
Okay, good. Have I said anything wrong yet? Let's see. Okay, so let's this is an interesting question. Let's say many viruses are quick viruses, and they're quick viruses in the sense of their genome might be sense single-stranded RNA. So if you package sense single-stranded RNA into a virion, and you get that into a cell, it can immediately make proteins. How would the virus integrate into a genome with this? Can you do that? What needs to happen?
It's a double-stranded break. Just kind of put yourself in.
And then RNA can just integrate into DNA?
No, it needs to be converted over to DNA.
Yes, it needs to be converted. So again, like my point is viruses, all about virus biology is all about like these weird interconversions between different strands and different directions of nucleic acids. So this would not be able to integrate into a lysogenic phase. It would need to get made, translated, or not, translated is the wrong word. It would need to get converted into DNA by some reverse transcriptase. And then once it's DNA, it could get inserted, integrated into the DNA of a genome. Make sense? Okay. Any questions on the life cycle before I move to the next thing? How much time do I have? 20? Okay. Gotta move fast. Okay. Let's just talk about sort of like virus sizes. This is a good thing to always keep in mind. So what's an ORF? An open reading frame, that's essentially like a gene that codes for a protein. What's the size of the smallest bacterial? How many ORFs do they have? 300. So the smallest bacteria, which is a mycoplasma, that's self-sufficient, can grow itself, has about 300 genes. That's the lowest level of complexity of a autonomous, independent organism. What is the size of the smallest virus? How many ORFs? It's less than 10.
Yes,
It's essentially, it's less than 10. I don't know exactly, but it's essentially like, it's less than 10. So most of the flaviviruses are something on the order of about 4 to 10 ORFs. So many of the viruses that we care about in MedVet, they're extremely simple biological machines. And there's only like 4 distinct things moving parts, you might consider them, that do stuff, okay? What is the most complex virus? What's about the size of the genome? So instead of orfs, we'll switch. So let's say this amount of information can be encoded in about 3.2 kilobases. So the size of perhaps one of the smaller viruses is about 3,000 base pairs of nucleic acid sequence. Something like a larger virus is on the order of 2.5 megabase pairs, which is 2.5 million. That's double the size of like a symbiotic bacteria. So if you think of like Wolbachia, which is what I study, that genome is about 1.2 megabase pairs, 1.2 million base pairs. There's viruses that are double the size of that bacteria in terms of genetic content. So virus, again, like it's sort of like if a virus is encoding that much information, can you really say it's like not alive? Like that's kind of bogus to me. If it's encoding double the information that a bacteria
Is that a
encodes.
virus that's like co-evolved? with the
What?
host? Is that a virus that would like co-evolved with a host, like a bacterial host?
I don't know, like why would it have, I mean, all viruses are gonna be co-evolved with their hosts, period. I think these viruses are gonna be more versatile and their life strategies are probably gonna be more niche, like more difficult. And they need to be more versatile so they have more genetic information. But that's always a trade-off because these viruses are gonna replicate way faster and evolve way faster. So just as this is like a brief sense of like the size and complexity, most of the stuff we will be talking about is tiny, like about 4 to 10 proteins. Any questions? Okay, so just to reiterate, the way that the genomic content is organized in a virus is important for its classification. So there's DNA viruses, which guess what their genetic information is encoded in? DNA. And there's double-stranded DNA viruses, and there's single-stranded DNA viruses, and there's single-stranded RNA viruses that are plus, and there's, which means sense, that means plus sense. There's single-stranded RNA viruses that are negative sense, which means it's antisense. I guarantee you there's probably double-stranded RNA viruses somewhere where they probably have a sequence that binds to another sequence. Is that correct? Probably.
There's single-stranded ambisense, which is both positive and negative.
Ambisense, yeah, so that would be ambisense. So it's like one of these and one of these. And do they ever bind?
So in ambisense, the positive gets regularly like transcribed, but then for the negative, they have to make, it's like what you showed earlier, when
Yeah.
it's a viral complement RNA.
Yeah. So essentially, like, if you're thinking about like, what do I need to take home from this? You need to take home that nucleic acids have direction, and they have there's differences. There's DNA, there's RNA, there's plus, minus, and viruses can essentially be like the whole gamut of those things. And what they are determines sort of like where they're classified. Okay. Let's see. Okay, so how do viruses assemble? So assemblies. So imagine you're in a cell. Okay. Imagine you're a virus that encodes like 4 proteins. Let's say you have a capsid, a polymerase. What else would you want? Oh, envelope protein,
Maybe a movement protein.
and maybe like a movement protein. Okay, so let's say like you're a virus. This is just imaginary virus. I'm just making this up. Let's say you got like 4 proteins. How do you assemble? If you only have 4 proteins, so if you imagine if you're like building a building, you have all these different workers. You have like pipe layers, you have like electricians, you have sheetrock people, you have concrete bricklayers. So there's a vast sort of infrastructure to build a building. How does a virus build itself with just these 4 things? Make a guess, a hypothesis.
Could it, like one be like multi-directional promoters?
You're saying some of these things, maybe what you're trying to say is some of these things might do more than one thing. That's a good way. That often happens. So sometimes we have what's called moonlighting, where the code of viruses is so compact that sometimes the protein learns to do multiple things. So they might have something that does multiple things. That's true. That can happen. But how else?
Doesn't it normally like hijack host machinery?
It can use host machinery. So in some cases, like there's different mechanisms of assembly. In some cases, it's going to rely on host machinery. But in other cases, it's actually way more simple than that. It's the capsid, the capsids always form these like nice geometrical structures, where if this is like one capsid protein, they sort of just naturally like come together. And you can build that into a molecule if you build sort of like protein-protein interactions. So let's say you build one of these molecules and it's less stable by itself, but it's more stable when it finds like a partner. Then it's gonna spontaneously assemble into that. Does that make sense?
You need to assemble the polymerase first.
Well, that needs to get replicated, yeah. So the polymerase would need to essentially replicate the genetic sequence. I'm more talking about how the structure of the virion, the capsid assembles. It's essentially like you can build, if the pieces are more comfortable binding to other versions of themselves, you can build structures that just autonomously assemble into these structures. And then what you bring up is an important point, which is after, or perhaps before, or perhaps during the virion is forming, how do you get the next replicated nucleic acid in there? In some cases, the virion assembles like around, so you might have a capsid that binds to like the genomic sequence, and then it sort of like assembles a structure around itself. In other cases, you have like loading where the virion assembles and then a protein grabs the nucleic acid and like feeds it in. Just understand like there's different ways to do it. Let's see. Okay, how much time do I have left? Like 10 minutes? 13, okay, that's good. Okay. Names of things that we're going to be talking about. Arboviruses. What are arboviruses? You had the perfect definition.
Arthropod-borne viruses. They are not a taxonomic group of viruses. They are more environmentally related viruses.
So so not taxonomic. That's
taxonomic because they comprise more over the 200 different families of viruses.
Yes, and so arbovirus is sort of like this broad term. It's like kind of like a colloquial term. What it means is arthropod-borne viruses. So these are typical viruses. So these are typically the viruses, right, that we're interested in because they are transmitted by vectors, the arthropods. So arbovirus means arthropod-borne vectors. It's a colloquial term that a bunch of viruses fit in, a bunch of diverse viruses that are taxonomically different. So that's important. That's good. Correct. Now let's talk about some actual taxonomic viruses that we will talk about. So again, virus taxonomy is super complex. I'm definitely not an expert on virus taxonomy. We're essentially going to be talking about alpha viruses, flaviviruses, and Bunya viruses. These all have about like 10 proteins. Most of these most of the arboviruses are chunked within these. So let's go through the characteristics of them. Okay, so Bunya viruses. Bunya viruses are segmented genomes. So there's typically about 3 segments, and they are single-stranded RNA segments that form closed loops. So if you have a virion of a Bunya virus, there's typically 3 different chunks of that genome, and oftentimes those chunks form what's called a closed loop, where if you have RNA can kind of like fold back in on itself if there's hydrogen bonds. And if these hydrogen bonds form, you can get either like a hairpin, or if it's like a giant hairpin, it's like a closed loop. And then there's oftentimes like structural, important twisting kind of things. So if you look at Bunya virus genomes, they kind of look like they're like braided and like tied in knots. So they have these 3 different pieces. Okay, so that's characteristic of Bunya viruses. They are enveloped, and they have what's called a polyprotein. What's a polyprotein? So in viruses, this is a common thing in viruses. they might, let's say I say they have 10 proteins. Sometimes they have less ORFs than 10. So imagine you had, say 9 proteins. You could perhaps encode 9 proteins in 3 separate ORFs. So imagine, this is just, I'm just making this up. Well, I'm not making it up, like it could happen. Imagine you had these 3 segments, okay? And so you have 3 segments, 1, 2, and 3. And let's say each of these 3 segments of the genome, it's single-stranded. Is it plus? I think these might be ambisense. Somebody fact check it.
The M and the S strand are ambisense. The
Ambisense.
polymerase is not ambisense.
So some of these sections are plus, some of these sections are minus.
Yes.
Very good, thank you. So a polyprotein would be, imagine you had one of these sense, and there was a start codon here, and a stop codon here. You could encode what's called a giant polyprotein. So imagine if this thing got taken by the ribosome and made into a big protein, maybe it looks like that, something like that. And then imagine if a scissors or a protease came and cut that in 3 separate spots. Then all of a sudden, how many proteins would you have? Well, I guess if you cut it in 2 spots, you'd have 3 proteins. So that is what's called a polyprotein. A polyprotein is when you have 1 gene, but a protease can cut that protein, and it gets essentially cut into 3 separate proteins that do 3 separate things. But it can be all encoded in 1 gene.
Where's the cut? When are the cuts induced? Since you're
Well, it'll get translated, and sometimes the cuts can happen like right at the ribosome.
Okay.
But probably like when the cut happens is evolved for each different virus differently. Does that make sense? So we understand what a polyprotein is. Okay, that's common in viruses, and flavies are the same way. You see these polyproteins. Okay, so these are characteristics of Bunya virus. Let's put for ambisense. Flaviviruses, 1, 2, 3. They are single-stranded RNA plus viruses. Okay. They are also encoding polyproteins, and they are also enveloped. Alpha viruses are within Toga Viridae. So there's Bunya Viridae, I think Flaviviridae, and Toga Viridae. So I should have said Toga Viridae. They are plus single-stranded RNA, and they are enveloped. Okay, examples of most famous alpha virus.
Triple E,
Oh, Eastern equine encephalitis.
Venezuelan and Western, like West also.
Oh my gosh, there's a bunch. Okay.
VEEV,
Wait, did you say VE?
Venezuelan equine
Encephalitis.
virus.
Yes. Okay, so we got these, and then chikungunya. Examples of flaviviruses. These are easy.
Dengue,
Yep. Dengue,
Zika,
Zika, yellow fever.
yellow fever.
These are the most famous
St.
ones. St. Louis.
St. Louis encephalitis virus.
You already know everything. Okay, Bunya virus. Examples. One famous one is Crimean Congo hemorrhagic fever. I don't want to write that out. Crimean Congo CCHF. I think that one's actually a tick-borne one. So, how much time do I have left?
6 minutes.
6 minutes, okay. Interesting thing about arboviruses, some can be spread by ticks. Most are spread by mosquitoes. So one reason why we focus a lot on mosquitoes in this class is because in a broad sense, mosquitoes spread like most of this bad stuff. Although there are plenty of viruses that like, I wouldn't say nobody cares about, but that other people don't necessarily care about because they're spread by like weird things that don't affect humans. Keep that in mind. The other really interesting thing about arboviruses in general is the taxonomy of the viruses matches the taxonomy of the vectors. So when I say that, do you understand what is the implication of that? If I say So if you drew a phylogenetic tree of the viruses, let's say virus 1, 2, 3, 4, and then you drew a phylogenetic tree of the reservoir, or not the reservoirs, the vectors of those viruses. The vectors of those viruses, the evolution, essentially the trees like match up. What is the conclusion when you see this? They co-evolve, and because they co-evolve, you don't get a lot of like crossover. Like that can happen in viruses, but this shows that these are highly specialized interactions. And the one reason that the arboviruses are super unique is that arboviruses are bizarre in that despite the fact that they're co-evolved with their host, they can also infect human cells in most cases. That's why we get sick from them. And that is not an easy thing to do as a virus, because imagine you have to learn and evolve how to infect a human's or insect cell. That's part of their life cycle. But then to also be able to cross over, infect a human cell, which is totally completely different from an insect cell, that is hard to evolve. So in that sense, the arboviruses are quite unique. Yeah.
Bilayer viruses infect plants too.
Yeah. So all I'm saying is, I'm not saying they only infect this or that. I'm saying that ability to cross over, infect multiple things, which typically causes disease, that's rare and that's hard to evolve.
A whole kingdom.
A whole kingdom?
Yeah, crossing over kingdoms.
Oh yeah. Oh, I see what you're saying. Yeah, a kingdom crossover event. Although if it's insect and plant animals, or plant and animal is even harder. Yeah. So that's a hard thing to evolve. Okay. Okay. We will wrap up. Final thoughts, and then the next lecture will sort of be some case examples of these viruses. Modes of transmission. Let's just go over some of these different modes of transmission of viruses. Venereal, what's that? Sex transmitted, and that can be either human to human or it can be like insect to insect. So that was one of the concerns with Zika. Was I think they found that it was human transmitted via sex. You're laughing, is that correct?
I thought you were gonna say it could be human to insect.
Not yet. Maybe in the future. Now you got my mind like visualizing what that process would involve. But my point about the venereal transmission is it can be human to human or it can be mosquito to mosquito. Like if you had how would a male get a virus? There's some viruses that can be, okay, let's actually, before I go to that, let's do vertical. What's vertical transmission?
Placenta, placenta.
Yes, or essentially like parent to offspring. And that can be, so if it was in placenta, that would be sort of like for mammalian. But it can also be in insects. It can be parent to offspring. So imagine if you had a female, I'm not saying Zika does this, but imagine if you had a female mosquito that had some virus that could do vertical transmission and then perhaps passed it to a son insect, and then that son mated with another female insect and it was passed through venereal. Like you could see how you could get these bizarre transmission interactions. That's my only point.
It will be like transvarial
Transovarial?
or transstadial, more transovarial.
Transstadial, let's talk about transstadial. Hang on. Transstadial, that has a specific term in insect terminology. What do you mean? What do we mean by transstadial?
Like goes through the stages of development.
Yes. So for insects, that's like sometimes if you get a virus, can it go through pupation? Or can it go through metamorphosis? Or can it go from larvae to adult? Or if it's a tick, can it go from a nymph molt to an adult mole? Some viruses can, some viruses can't. That's transstadial transmission. Horizontal. What's horizontal?
Between other individuals.
Or potentially different hosts. So horizontal could be you get it from a bird and then the mosquito transmits it to a human. Or individual to individual, perhaps like in the case of if in dengue, a human has dengue, and then a mosquito bites it, picks it up, and transmits it to another human. That's horizontal transmission. And then there's mechanical. Mechanical is like the dirty needle, like HIV/AIDS. That's mechanical transmission. Aerosol. Aerosol is like, it gets in your lungs, you cough it up, and it's in the air. You can breathe it in like COVID. Just understand that there are a vast diversity of transmission mechanisms in viruses. Arboviruses can do a mixture of some of these depending on the virus. Okay, so that's my basic virology 101, and then next time we will talk more about some specific arboviruses. And throughout the rest of the class, we will go more in depth into specific arboviruses. Good?
Yeah.
Okay, cool. Okay, good work. We're done.